The science of genetics is concerned with the study of how traits are passed from parent to offspring. Genetics has a cellular basis that is rooted in the processes of mitosis and meiosis. Mitosis allows for asexual reproduction, and meiosis ultimately results in the production of gametes that fuse to create offspring that are genetically unique. That's sexual reproduction. I expect by now you have read chapters 9 and 10 in the textbook and understand the processes of mitosis and meiosis and watch the videos. Now we're going to move into a study of genetics on a more organismal macro level, tracking traits through multiple generations, learning how we can make predictions about the outcomes of a genetic cross. We're going to begin with looking at the simplest modes of inheritance. We call them Mendelian traits, and the name Mendelian refers to Gregor Johann Mendel, the individual that we attribute to being the father of genetics. Now, like the father or mother of any discipline, Mendel was the first person that we know of to clearly articulate a model for how inheritance works. Now, it's a bit of a play on words also to call him the father of genetics, because he was actually a priest. Technically, he was a monk in the Augustinian monastic branch. He had a successful career as ultimately the abbot of his monastery. But like monks in his day, he was intellectually curious, and he had hobbies that occupied his mind, and his was the study of genetics. Mendel actually used scientific methodology as we have described it, where he asked questions, he conducted experiments, he published his research. He published 2 papers in his time, and the one for which he is known was called Experiments in Plant Hybridization, and it was published in 1866. Now, his paper went unnoticed for about 30 years. And there's a few reasons why the larger biological community did not really appreciate what Mendel was saying. For one, it was published in a rather obscure journal called the Proceedings of the Natural History Society of Brunn, which is in the Czech Republic. Also, it was published in German language, and most of the research in genetics in the late 1800s was happening in English-speaking countries. And Mendel was proposing ideas for which no cellular framework was known. So he was definitely a bit ahead of his time. Mendel's research involved utilizing peas as his model organism. So one of the first things we did in this course was talking about how model organisms are very useful for asking and answering questions. Peas turned out to be pretty good model organisms for genetic research. First of all, they're easy to grow. Mendel grew peas in a small walled garden on the grounds of the monastery. Peas grow up, they like to climb, they have tendrils. So because they grow vertical, you can fit lots of them in a relatively small space. Once they germinate, they do pretty well. A second characteristic is that they have a pretty short generation time. Peas are what you'd call a cool season crop. I just planted my peas a week ago. So they will produce 2 generations in a season, one in the spring and one in the fall. Here's a really important characteristic. Peas have many contrasting physical traits that can be used as markers for tracking the inheritance through generations. Here we see a list of some of the variable traits in the peas that Mendel grew. Things like flower color, seed color, the height of the plant. So Mendel would create what he called true breeding strains, maintained true breeding strains, beds of peas where they make purple flowers, and when you cross them with each other, they all make purple offspring, and other beds that had white flowering plants, and you cross them and they always produce white flowering offspring. We call these traits phenotypes. Now finally, one of the most important things about peas is you, the researcher, has control over pollination. Pollen is the source of the sperm cells in plants. So when you transfer pollen, you are providing the sperm that will fertilize the eggs in the flower. Most plants will not self-pollinate. Peas will. They have a complicated flower structure. The male and female parts mature at the same time. And if you leave a plant alone, it will self-pollinate. That makes it very easy to inbreed and maintain what he called true breeding strains. You also, though, have the ability to cross-pollinate. So this figure shows the basic technique. If Mendel wanted to cross purple and white flowering plants, he would start by removing the pollen-producing structures from the purple plant and physically transferring pollen from a white plant to the purple. Now what we're going to do is look at an example of Mendel's research that involved a single trait, look at what he did and how he interpreted that. So this first cross involved a true breeding purple flowering plant crossed with a true breeding white flowering plant. This first initial generation is what Mendel referred to as the parental or P generation. The generation produced by that cross, the first generation of offspring, is called the F1 generation. F stands for filial, meaning pertaining to siblings. So here we have a generation of siblings that come from the same parents. When the F1 seeds were planted, they all grew into purple flowering plants. So this established an important but often misunderstood Mendelian concept called dominance, that the purple trait is dominant to the white trait. Now, the next step would be to cross 2 of those purple F1 with each other. So here in this figure, we see the transfer of pollen between plants, the production of the seeds, planting those seeds. That's our F1 generation. Now, here's the results Mendel obtained from crossing the F1 plants. That would produce a second generation of offspring, an F2 generation. And when all the seeds were planted and the plants matured and he counted them, he got very close to what we would refer to as a 3-to-1 ratio. 3/4 of the F2 plants produced purple flowers and 1/4 produced white flowers. So this figure in chapter 11.3, gives us some actual data from Mendel's paper. And visually we see the P generation, the F1, right? The results of that cross. They're all purple flowering. Purple is the dominant trait. Cross to purple F1s, and 705 of the offspring were purple flowering and 224 were white flowering. So if purple is the dominant trait, why does white pop up again in the F2 generation? What the heck is going on there? So Mendel explained this, proposed a mechanism for this that involved mathematics. So what we're going to do next is look at Mendel's interpretations and kind of put that into the context of what we now know about inheritance. All right, so how did Mendel explain his observations? What were his interpretations? Mendel made 5 assumptions. He basically said, I can explain these data if the following things are true. Number 1 was that each trait, like the flower color or the plant height, is determined by a different heritable factor. Right? So that's a translation of Mendel's term, a heritable factor. Today, we call those heritable factors genes. That's not a term that was in use at the time. DNA was not known to be genetic material. Chromosomes, what's that? So Mendel was imagining some kind of physical particles that were passed from parent to offspring, and he called those things heritable factors, right? So each trait he looked at was controlled by a different gene. A second assumption he made is that the organisms involved have a pair of factors for each trait. We know today that peas and humans and most eukaryotic creatures are diploid, that our genes are found on chromosomes and our chromosomes come in pairs. So Mendel was predicting this. This whole model would only work if the individuals had a pair of genes for each trait. A third interpretation was that although an individual has 2 factors for each trait, there may be different versions of that factor out there in the population. And if alternate versions of a factor exist, one version is dominant to the other. So we use the phrase dominant and recessive to refer to the alternate versions of a gene. Today we use the word allele to talk about the alternate versions of a gene. We have a dominant allele and a recessive allele. Mendel proposed that as the parent organism is making the gametes, that these pairs of factors separate, or as he called it, segregate Mendel's law of segregation is the idea that the diploid parent undergoes a process today we call meiosis. Those pairs of factors separate or segregate so that the gametes are haploid. They only have one allele from each pair originally present in the parent. The final assumption is that when fertilization happens, the combination of gametes is random. It's random as to which sperm is going to fertilize which egg. But when fertilization happens, it brings those pairs of factors back together in the embryo that we call zygote. Zygotes are diploid, right? So when two haploid gametes fuse, it produces a diploid zygote, which then grows and develops through mitosis to make a multicellular diploid individual. So Mendel made some pretty remarkable predictions without ever having studied meiosis, never seen chromosomes, never hearing what DNA was. So this is figure 5 in chapter 11, where Mendel's cross is shown visually, and it's tracked by using symbols for the alleles. The tradition is to use an uppercase letter to represent a dominant allele and a lowercase letter to represent a recessive allele. Since the parental generation was true breeding, they would have 2 copies of the purple allele in the purple parent and 2 copies of the recessive white allele in the white parent. We say that those parents are homozygous, right? Homozygous is a condition where you have 2 copies of the same allele. So what he did was cross the homozygous dominant purple plant with a homozygous recessive, a white plant. So the flower color itself is what we call the phenotype. The combination of genes is what we call the genotype. There we see the possible gametes that could be produced. Homozygous individuals can only make one kind of gamete. Gametes are haploid. The purple flowering plant would produce all gametes that had only the purple allele. The white flowering parent would produce gametes that only contain the white allele. There's only one possible genotype that could be produced in F1, and that is to have one each of the 2 alleles. We call that the heterozygous genotype. And because of dominance, heterozygotes have the dominant phenotype. All the F1 would be purple. Now when the F1 produce their gametes, when those pairs of alleles separate, there are 2 different kinds of gametes that could be produced, and we expect a 50-50 mixture of those 2 kinds of gametes. Half the gametes should have the dominant purple allele, half the gametes should have the recessive white allele. Now when you cross 2 of those F1s, we have 2 different kinds of gametes made by 2 parents, providing us with the opportunity for 4 different combinations in the offspring. So we see that the combination of the 2 dominant alleles would produce a homozygous dominant offspring. The combinations of one purple and one white allele would produce heterozygous offspring, and the combination of two recessive white alleles would produce white offspring. Now, when we're reporting the result of a cross, we typically express the outcome in terms of ratios. And you can have two different ratios: a phenotypic ratio, so i.e. what do the plants look like, and a genotypic ratio, what combination of alleles do they have. So in Mendel's F2 generation, we saw a 3-to-1 phenotypic ratio. 3/4 of the offspring would have the dominant purple phenotype. 1/4 we would expect to have the recessive white phenotype. The genotypic ratio is a little more complicated. There are 3 genotypes possible: homozygous, heterozygous, and homozygous recessive. So there we have a 1-to-2-to-1 ratio. So 1/4 of the genotypes would be homozygous dominant, half the genotypes should be heterozygous, and 1/4 should be homozygous recessive. When you have a heterozygous individual, it has the dominant phenotype. So that means individuals with the dominant phenotype can be of 2 different genotypes, whereas the individuals with the recessive phenotype, we know they must be homozygous recessive. So that's an important known in genetics. If you have an individual with the recessive phenotype, you know what its genotype must be. Whereas if you have an individual with a dominant phenotype, unless you know exactly what their parentage is, you don't know for sure what their genotype is without doing some experimentation. Now finally, to put this into a more modern context, we have this scheme here. Scheme sounds nefarious. This model for how inheritance works. Those heritable factors Mendel talked about we call genes, and genes are sequences of DNA. And genes are the directions to cells for how to make proteins. The nucleotide sequence of a gene dictates the amino acid sequence of a protein. And it's the presence of proteins ultimately that produce traits. A phenotype is a color or a shape or the ability to perform a metabolic process, and those are due to the presence of proteins, and the proteins are the result of gene expression. Now in our peas, we saw that there were 2 traits, purple and white. The original condition in peas is to have purple flowers, but at some point a mutation occurred. We think of mutation as being bad, but mutation really just means a change. New alleles arise occasionally as existing genes experience changes. So we'll have a new nucleotide sequence that produces a slightly different protein that can result in a variation in the trait. In the case of the flower color trait, we know what gene it is, right? We found the gene on the P chromosomes. We know that each gene has a specific place on a chromosome where it's found. We call it the locus of that gene. So if we look at the locus here for flower color, and we see there's a certain DNA sequence that produces a protein enzyme that catalyzes a chemical reaction and results in the production of purple proteins. But the white flower allele, there was a mutation. In one particular spot, there was a nucleotide change, and that results in an absence of that enzyme. So without that enzyme, that chemical reaction never happens, and the flowers are white instead of purple.